Thank you for submitting your protein booklets. And if you have not already looked at the slide presentation and done the writing assignment and the quiz that goes with that, go ahead and stop this and get that done first and then come back to this. So this video is going to encompass part of protein synthesis, or how proteins get made. So protein synthesis is broken into two parts: the part that happens in the nucleus and the part that happens outside of the nucleus in the cytoplasm. And so in this video, I'm going to just go over the first part, which is called transcription, where messenger RNA is assembled in the right order to reflect the code that's on the DNA. So we already went over this right before spring break, so it should be review for the most part, but I will add in a few new words, and also there are different notes up on the board now than what you went over before. I still have those old notes, and I'll try to remember to flash over to those at the end of this. And if you still have your journals with you, you can go ahead and keep some notes in your journal. And I guess let's get started. Okay, so not all of your DNA codes for genes. Codes for genes, codes for proteins. The part that codes is called a gene. And not all of your DNA is coding. So you've got long strands of DNA, and you've got 23 pairs of those, so 46 long strands of DNA that will coil up into what we consider chromosomes. We'll talk about that later. But not all of that DNA material actually codes. So there's non-coding and coding parts. To recognize the coding part of the gene, there's actually kind of a message before the code. And so the message is in the form of a ta-ta box. So ta-ta box is a sequence of T-A-T-A-T-A to let you know that a gene is on its way, to let the RNA polymerase that's going to open up the coding area of the gene know that, hey, a gene's coming. So before I go on any further, I've got a few notes on DNA here just to refresh your memory that DNA has a complementary code. So the two strands of the double helix reflect the nitrogen bases and their complement. And I don't know if I've used the term complementary code yet, but get used to that idea, get used to that terminology. What complements with what. So we talked about pointy letters complementing with each other, so T and A, and then C and G, the round letters, complement with each other. And so these complements are held together with the weakest possible bond in an aquatic situation, and that's going to be a hydrogen bond. So the T and A's are held together by two hydrogen bonds. And we had represented that by taping them together with thread. And the C's and G's are held together with 3 hydrogen bonds. Now, the rest of the DNA needs to stay sturdy, so it's held together by covalent bonding. So we represented that with the tape, that this shouldn't come apart. So the rungs of the DNA shouldn't come apart. And then also, the nucleotide itself is covalently bonded. And if you remember the parts of your nucleotide, which is the monomer for a nucleic acid, we have the phosphate head, the deoxyribose sugar, and the nitrogen base. In this case, the nitrogen base is adenine. Okay, so covalent bonds there, and covalent bonds holding the rungs together. So at the site where the ta-ta-ta begins, following that, if there's a gene there, it's going to have a start codon, and the start codon is always the same code. I've got to remember to look at the camera and not the human. So, hi everybody, learning new skills. Okay, so we've got ta-ta-ta-ta-ta-ta, and then we have TAC. TAC is the code for the starting of a gene. So it's the start codon. Codon is referring to 3 nitrogen bases at a time. So throughout the whole, generally really long genetic code, it's going to be read 3 at a time, TAC. And then my next one I've written up here, codon to reflect GT. G is the next codon. And so we'll keep reading those. And again, what opens up this whole thing is RNA polymerase. When the DNA is opening up just a portion of itself in order to make a gene, that's done by RNA polymerase, which is an enzyme, which is a protein, as you know. And you'll recall that enzymes are proteins that get a job done without getting used up. So the scissors are a really good analogy because they could cut open lots of thread and not get used up. And so this code goes through until there's a stop codon. Now there's 3 different kinds of stop codons. So TAC is the only start codon, but there are 3 stop codons. So the stop codons I have up here, it's either going to be a TAG, a TGA, or a TAA. So TAG, TGA, or TAA. Those are 3 possible stop codons. So if we follow this 3 at a time, and it's really important that when you're practicing exercises with DNA that you are clear about those codons 3 at a time. You can't add to or skip. So we have our start TAC, then GTC, GAC, AAC, AGT, CGT, TAG. So that's a stop codon. So that would tell the RNA polymerase to stop opening that up. And then the next thing that happens, or is going to be happening as this is being cut open actually, is that nucleotides of RNA are going to start falling into sequence. They're going to match the complementary code of the DNA. So we've got to start with what goes with T. So what goes with T isn't this one, is, okay, well I have one, oh I see it, it's here. I have one here, but by the way, this one won't work. Why? Because it's got 3 heads. It's a triple-headed monster. Look at this though. This is kind of cool. So here's a RNA molecule. This is an RNA monomer, so this is a nucleotide of RNA, and it's got the phosphate head, and it's got a sugar, but it's the ribose sugar instead of deoxyribose. So RNA gets its name from the ribose sugar, and then it's got the green still represents adenine. It isn't labeled, but I'll put an A on that. So that's adenine. And then check this one out. It's got the adenine, it's got the ribose sugar, but it's triple-headed. This is adenine triphosphate. This is ATP. So ATP is the 3rd nucleic acid that we're talking about in class. So we've talked about DNA as a nucleic acid, RNA as a nucleic acid, and we've been talking for a while about ATP. This is what ATP looks like, triple-headed monster. So it's not going, it might be floating around in the nucleoplasm, could be, but it's not going to get used because it wouldn't fit into the puzzle. So let me put that back up. And so T and A go together, so adenine and thymine. So, oh, I got to put my heads the other way. I've got to have this side, my 5' facing the other way. So my 5' side has the smiley face going the other direction. So they're going like this. I don't think I did that when I met with you guys right before spring break. Okay, now this is another difference about RNA, which is it doesn't have thymine. So you can look around the board for thymine to put in, lock in place with that adenine, but you're not going to find it in a, hello camera, you're not going to find it on a RNA nucleotide. It doesn't exist on RNA nucleotide, but this does. This orange one that has a U on it, uracil, is going to fit in place. So this is a short nitrogen base. This is a long nitrogen base. And also it matches up with my pattern, okay? So it's going to have very much the same shape as a thymine, but not a thymine. Now, for our convenience, so you don't have to watch me struggle to try to find all the nucleotides. Oh, look, I already made a strand. Okay, and I need another. I may not be able to get to the end of the strand. I suppose we'll just call it. So it would make this all the way through to the TAG, and we'll just pretend we did that. Okay, so if we had just imagined that this was like the whole thing, and so this messenger RNA that is covalently bonded, and it has, oh, don't look over here, and is the whole gene, we would hope, is going to leave the nucleus and go over to, where's the protein coded for? Yes, the ribose. So this will leave the nucleus through a nuclear pore and go to the ribose for part 2, which will be called translation. So transcription, if you think about it, this messenger RNA, because it's the messenger that's going to get this code and take that message outside of the nucleus and go out of the nuclear pore and into the cytoplasm. It's a messenger, so we're going to call it mRNA, and it scribes, or it's like it's writing down the code in its own language. Okay, but it scribes the DNA code, so we call this transcription. I had asked some questions related to this. I just want to go back over really quick. So it should be starting to make more sense to you because I'm going to be using this terminology. So just make sure that this sounds true to you. Deoxyribonucleic acid is deoxy, meaning it has fewer oxygen molecules than ribose. So just that words mean things. That's how it got its name. Okay. It's double-stranded. This macromolecule, it's a big molecule. I mean, macromolecule means it's made of so many different parts, lots of parts, okay? And so it might be repeated parts, but it's like it's a big molecule relative to, you know, this phosphate's a molecule too, but it's a tiny little thing. This is complex, so that's why macromolecule. So this is a double-stranded macromolecule Ribonucleic acid has the ribose sugar instead. It has 2 more oxygens than deoxyribose has. It has uracil, not thymine, as a nitrogen base. Actually, this would be good to know. I'm jot it down. It is single-stranded, and it will helix. It will twist on itself, but it's always going to be in the single strand, and it's also going to be really a ton shorter than the DNA because it's only going to be as long as 1 gene on your DNA, and your DNA has hundreds of genes on it. Okay, and so it's either going to, this is like it'll only be as long as the gene, and once we get into the cytoplasm, there are also other kinds of RNA called ribosomal RNA and transfer RNA, and we're going to talk about those in the next video. Now, just quickly, one thing that we did go over before we parted that you recognize as a really big deal now is we talked about how viruses can hijack the system to get their own business done. Now, for coronavirus, the hijaxing doesn't occur in the nucleus. It's actually going to be happening in the cytoplasm. So from a virus's standpoint, that means they don't have to go through double nuclear membranes to get their business done. They don't have to go to the big boss. They're going to get everything done just sneaking in to the cell and going straight to the ribosome. So one thing that is relevant to this part of it, and this is what I talked about before we left, was that, so viruses will either have DNA code or RNA code, and the coronavirus consists of RNA code. All right, so it taped upon itself, and then some of it tore off. Oh my goodness. So it's going to have long stretches of RNA code that has its message on it. Looking at the camera. And so next time we'll talk about how the virus invades the cell and uses the ribosomes to make more of itself. But in the meantime, I'm going to go ahead and post the same video that I had up since I partitioned you all out into separate classes on Google Classroom. You don't have access to that video. I don't think I archived that old one. So I'll go ahead and put up the video so that you have access to it. Whoop. Okay, until next time, thank you for watching me struggle with my first video. I'd do another take, but I'm tired. It's time to stop. So be well. I'm thinking of you all. I miss you terribly. And next time, ta-ta.